Good morning everyone and welcome to this Cryo-EM course. So last class we are discussing about the lens hysteresis and how it is going to affect the imaging, but that time I didn't draw any diagram to explain it what I am trying to say. So we will discuss about those things also, and that will help us to understand the system. Before that, last class we discussed about that EM lens system. So what is that EM lens? So always remember that EM lens system, as I mentioned, there will be a deflector. So that is called, this is a deflector. Then there will be a electromagnetic lens. If we just consider that, okay, so we all know that electromagnetic lens, it will look like this. So there will be a gap, those will be the copper wind properly. So here there will be a gap between these two, so those will be the copper winding, and this will be the gap where the magnetic fluid will come, and then there will be a two lens system, that is called the stigmator, STIGMA, stigmator. The stigmators will be there, and then at the end this is our optical axis, and at the end we have a aperture. So aperture is a small disc which is going to block the more deviated electron beam. If something is close to optical axis, it will help us, so it will be aperture. Okay, so this is the entire lens system. Whenever I am talking about condenser lens 1, condenser lens 2, then objective lens. So objective lens will be split into two half, upper and lower objective lens, and then intermediate lens, and then projector lens. Okay, so the projector lens again 2, it is 1 and 2. So sometime in between the condenser lens 2 there will be a condenser lens 3 will be also there. So we mainly consider this. Now all the lens system is connected with these things, that we have a objective lens or condenser lens 1 or condenser lens 2, it does not matter, it always has a two deflector, and then there will be electromagnetic lens, there will be a stigmator, and there will be aperture. So aperture, I told you there will be a hole inside. So this is block, this is a metal disc, this will be blocked, and this is the hole where electron beam can go. Now we can use the different types of magnetic field to converge or diverge the electron beam. Now what is the role of the deflector? Sometime electron beam is not coming through the axis, it is deviated from the axis. If it is deviated from the axis, so if I just use different color here, so if you just consider that my electron beam is coming from this direction, it is not following the optical axis. So then definitely most of the electron beam is going to block here, and that is not a very ideal scenario, right? So now here we are using a deflector, some kind of magnetic field, which it will help us to deviate the path of this electron. So electron cannot go straight, we are using a magnetic field here. And then again this one, we are using a certain kind of magnetic field which it will help us to not only deviate, follow this path, optical axis path. So it will come in this way and try to push in this direction. So if there is a deviation of that electron beam in one direction, or if it is deviated from the optical axis, this deflector plays a very vital role to bring the electron beam through the optical axis. So that is the reason deflector is always we use it during our microscope alignment, and it is very important when the electron beam or light source it will come, always it should follow the optical axis, and this is our optical axis. Now, so this is the role of the deflector. Then stigmator also, it is helping to arrange the beam, and it should follow this optical axis, and it should follow a circular way. All the beam should come as a parallel. Okay, stigmator is dealing these things. Now there will be always some kind of aberrations will be there. If we are going to discuss all this aberration, there will be 4 major aberrations are there. One is astigmatism, second one is the spherical aberration, chromatic aberration, and coma. So those things we are going to correct with this astigmator or the deflector. Now what I was talking here, that most of the time our electron beam is going to block through this aperture. So now if you just consider, there is another electron beam is coming in this direction. Okay, so they are coming, there is a magnetic field, there will be a deviation, and there will be a crossover point somewhere here. So if we just consider that, okay, let me draw these things little bit proper way. So if we just consider that, okay, first there will be a magnetic field, and then if this is the ray, it is coming and converge in particular point. Okay, so when they converge into particular point, if they will just follow the path, right, they will follow this path, and in this particular case there is all over there is a beam. It is not like that only 1 or 2 beam is going to come. So in this particular sense you can see this is the area where it is allowing some of this electron, and rest of the electron whatever it is coming through this, it is going to block all the electrons. So that is the role of the aperture, that if some electrons are deviated too much, it is going to block by this aperture. It will allow only certain electron beam which are close to the optical axis and very coherent, and that is going to allow through this aperture. So this is the overall system of a electromagnetic lens system. Now let us see in the microscope room. If we enter to the microscope room, do we always see that it is written as a condenser lens system? It is a condenser lens 2, objective lens, or projector lens, or intermediate lens. There is some specific name of those lens systems. So it is better we should know which lens is connected with what. And deflector, as I mentioned that, okay, there is a deflector, this is the common lens system for all these lenses. For the condenser lens, these 4 items are there. If we just consider, this is item number 2, this is item number 1, then 2, then 3, and then 4. All these 4 items will be there in all the lens systems separately. For condenser lens 1, condenser lens 2, objective lens, like that. Now if we just go to the microscope room, so there will be a beam deflector, there will be a gun deflector. So there is a different whenever it is connected with one particular lens. So those things, those deflector or stigmator is connected with either objectives or condenser lens. So all these lens systems has these 4 components, but it has some separate names. So it is better we should know those lens system, that okay, what are those lens and how we can use those things. So we are very much familiar with this setup, that okay, how those things are connected. I already showed you these things. So this is the entire microscope, and then condenser lens 1, condenser lens 2, objective lens. This is a 2 split, that upper objective lens and lower objective lens. And previously we also connected all of this with here, that okay, the gun is connected here, our accelerating stack is somewhere here, then this is our goniometer or sample insertion area, this is our condenser lens aperture, then objective lens, and all the projector lens are here. Projector and intermediate lens are connected is here. This is the viewing chamber which is connected with this one. Our camera will stay at the back. Nowadays we are using a mainly for biological science sample purposes, we are using K2 camera or Falcon camera. Most of the time those cameras will go at the bottom. So it is a side mounted camera, we are using CCD, or the bottom mounted camera mostly we are using this direct electron detector, either it is K2, K3 or Falcon. So this is the same column architecture. So now you guys are very much familiar with the gun deflector. So I mentioned that each and every lens has a different types of deflector. Whenever we are talking about the gun condenser lens, it is coming because electron beams are coming from the gun. So now you can see deflector is there, stigmator is there, aperture is there. So I splitted this entire system with these 4 component. So we have a deflector, we have lens, we have stigmators, and then we have aperture. Now same thing is true for the objective lens system also. So this is the objective lens area where we have a deflector, objective lens, stigmator and apertures. If you go down, you can see that image deflector is there and rest of the lens system is there. Now we start from the top. So we have the filament. We already discussed about this filament for a lot of time, and we have a different types of filament, that is tungsten filament, and as well as we have field emission gun or Schottky emission. So here we have a Winot cylinder which is covering this filament. So whenever we will go to the microscope room, we will see that it is written filament or current bracket, it will come as a filament, but you cannot see anywhere it is written the Winot cylinder. So Winot cylinder basically written as bias or emission. So you can see that this emission, it is going to control that, okay, so how much emission is there. Then in gun it will be a different type of gun or filament, whatever we want to use it. Now it is coming to the accelerating stack. There will be a multiple stacks are pile up together. So here the accelerator stack, it is called the voltage or high tension. So that is, we are showing you that in previous column that I mentioned that in this the high tension is connected through this one. Previously we mentioned about that 100 kb microscope, 200 kb microscope or 300 kb microscope. This kV is coming through the high tension. It is definitely not taking from the normal power supply. It is developing that 200 kilovolt or 120 kilovolt or 300 kilovolt, and it is taking that amount. Now it is coming to the gun. So now you can see there is a 2 magnetic system is there, electromagnetic system, which is called the gun deflector. If there is any changes happen in the gun region, if the electron cannot come through the optical axis or there is some kind of deviation, that gun deflector is going to correct that one, and that will be, you will get it as a gun shift or gun tilt. It will be not written as gun deflector anymore in the microscope room. Now the first lens system it is coming, that is called the condenser lens 1. So condenser lens 1, if you look at the condenser lens 1, it is mentioned as a spot size. So that is a very important parameter, that high spot size and the small spot size. So when you are using a high spot size, you are getting a more coherent, and low spot size means big and less coherent electron beam. We can discuss these things in the later stage with a ray diagram, and this will be clear to all of you, and this is a very important part for our biological sample purposes, especially for cryo-electron microscopy. We are using a very high spot size because we want to control our electron dose using this C1 lens, and so the spot size is an important lens system for our purposes, that is we are going to discuss in the later stage. Now it is coming to the next lens system. That next lens system, it is called the lens 2. It is a condenser lens 2, or we can call it is intensity. Whenever we will enter in the microscope room, we can see it is written as intensity. It will be not a condenser lens 2. We can see only condenser lens 2 it is written if we just want to correct their astigmatism. Astigmatism is a aberration. We will going to discuss about that. So, your beam will be elliptical, it will be not circular anymore. So, that time your stigmator is going to play an important role. So, here you have a condenser aperture, this is the condenser aperture, and then this two we can say that it is a condenser astigmator. So, there will be two kind of astigmator we can get it, that okay, the condenser astigmator which is going to correct the condenser astigmatism, size and the centering. We can control all of these things. Now it is coming for the next one, that is the imaging system, where we have the sample holder. That is, I was telling that the goniometer or the stage area where we can insert our sample, and the sample will stay in between these two objective lens. It will be upper objective lens and the lower objective lens. Now above this sample it will be a beam deflector, because beam is coming here. Now we are telling that this is a beam deflector. If you look at this beam deflector, you can find it in the microscope room, it is written beam shift or beam tilt. Now objective lens is connected, this is our sample holder, and here one more important part is there, that we have to insert the sample in one particular position. It should not change its position. So if we have a sample here and if we have a objective lens, so this is my sample and this is my objective lens. So upper objective lens, lower objective lens, and I am inserting my sample here. This is the only place where physically I am changing the position of something, right. So when I am changing rest of the lens systems are fixed in the electron microscope. So I am physically putting my sample or adding my sample inside this objective lens. Now here we have a particular position where my sample it will go and stay there, that is called the eccentric height of the sample. So if eccentric height it is changed, it is going to affect our rest of the imaging part. So that is the reason here position or z height or tilt is very important where we are storing our sample. So we are keeping a exact z height. If it is not, we have to adjust our z height, that we are going to discuss again later. Now objective lens, is it going to if can you see it is written as an objective lens? No, we cannot see it that it is written as an objective lens. You can see it is written as focus, because objective lens is a very strong lens. We already discussed this one, and this is basically helping us to focus our electron beam. And now whatever stigmator and aperture is connected with the objective lens, it is called the objective stigmator or objective apertures. Now if you come down of this one, you can see that it is next, it will be image deflector, which is appearing as image shift and tilt, and then there will be two intermediate lens system. So there is a projector lens and two intermediate lens and two projector lens, which can change the magnification from 5K to 22K or 32K. You can get a different magnification. So now from this one we can get the detector and get all this imaging information. So this is the overall architecture of the microscope, which is connected with different option, and their names are different. It will be not written like a gun deflector, condenser lens or condenser stigmator or condenser aperture. Now let us take a look how this TEM server or user interface look like. So it is very important, whoever is going to the microscope room, what kind of user interface we are expecting. So here you can see This is the TEM server or user interface. Many of you are familiar with these things, whoever is using electron microscope, because this interface specifically for FEI or Thermo Fisher microscope, this is the interface you can always see. So, geol microscope, this interface may be different. First thing, if you look at this main page, what you can see? You can see accelerator, column, autoloader. So, here this is showing the vacuum of your system in a log scale, which is very user friendly. We can easily see this number. So, basically if you change that, you will get it in torr or Pascal or in Ember. So, this is basically showing our vacuum system, that how good is our vacuum. So, here near the column and the accelerator or gun, we are always expecting a very high vacuum, because last class I discussed about that we are using either lab 6 filament or field emission gun or Schottky filament. All these filaments are very expensive. If this vacuum is not high, if there is a moisture or air inside that column, it is going to affect our filament, it is going to damage that filament, filament will be contaminated. And if filament is contaminated, if the source of the electron is contaminated, it is going to affect our entire system. That is the reason we are always expecting a very high vacuum in the accelerator and the column region. So, we are going to discuss about what kind of pump we are using for the vacuum. Mainly you just remember, for this high vacuum we are using the ion getter pump or ion pump. Now, here you can see it is written high tension. I already showed you what is called the high tension. Wherever there is an accelerating stack which is connected with our high tension value. So, here this microscope, it is a 200 kV microscope, and that is the written that is the reason you can see it is written as a 200 kilovolt. Now, there is a gun lens is there, and there is a extractor, that there is a certain voltage you have to hit that filament, and also that field emission gun emission current, you can see it here, and there are some other options are there. If you just want to go in details, you can get all this information. And whenever we are replacing a FEG or field emission gun, we have to store all of this information, that gun length, what kind of gun length value we are using, then what is the heating value we are using. So, those values we have to store here. And now, there is some button you can see it, that okay, it is written MFX, MFY. So, those are called the multifunction X and multifunction Y. We will come back and we will discuss about this, what is this multifunction X, multifunction Y. Now, this is the area where we can see our images, what kind of images we are imaging. Okay, one more important panel, if you look at this bottom panel, this is also very important. You can see the magnification here. What is the magnification? Currently, whatever magnification we are imaging, that is 45,000 x magnification. Okay, and here you can see it is written SA. So, this is called the SA mode magnification. One condition you should remember, that is one of the major difference for light microscope and the electron microscope. Electron microscope we are using electromagnetic lenses, and those lenses are fixed. It is not like light microscope objective lens, that we can just change the position of the objective lens. So, electromagnetic lens we cannot change the position, we can change the magnetic field, and that is helping us to magnify our images. So, you can start from a very low mag, even if you go to 50x mag or 75x mag, and then you can go to a very high mag. So, there will be a 3 different mode in electron microscope when you are imaging. So, one mode will be LA mode, which is a very low magnification imaging you can use. Then it will come to the medium mode. So, above 1000x magnification to 2600x magnification, it will be in LA mode. After that, it will convert to the SA mode, where mostly we are using the very high magnification imaging. Now for the biological science sample purposes, if our samples are very very small, so if we just consider that we want to image a protein sample, where the protein sample, the molecular weight, you just consider that it is a 300 kilodalton protein. So maybe one monomer or one protomer is 60 kilodalton and it has 6 subunit. So it is coming 360 kilodalton or near around 400 kilodalton, or if you just consider Cryo-EM. So all this sample you can go to minimum magnification, what we are showing you here, something that 45x magnification to 60x magnification, and that will be good enough to see those molecules. But this particular magnification, even you can see the DNA protein interaction. So there are some DNA binding proteins are there. So if you just want to image those things, you can use this similar magnification. Now if you want to image a virus particle, you can use the same magnification, or you can go little bit lower magnification to see more virus particle in this same magnification. Now if you want to image a bacterial cell or normal human cell, that macrophage or neutrophil, you want to image it, generally you have to go to a very low magnification. So for the bacterial cell, if you want to image, so sometime in some bacteria it will be good enough to that you can see, want to see the bacteria and virus interactions. So generally 20K magnification is good enough to see those kind of interaction and do the tomography also, that virus and bacteriophage and bacteria's interaction, and we want to get a 3-dimensional structure. Now I want to image a cell, so any human cell, macrophage. So generally we have to go to very low mag if we want to capture the entire cell, because any normal human cell, if you just consider the macrophage, neutrophil, RBC, sizes is almost near around 10 micrometer, and we have to go to low mag, so below 6000x magnification. Now the other hand, I want to image a very small protein, so that protein may be the G protein couple receptor with G protein, and the G protein couple receptor and G protein I want to image, definitely I have to go to a very high mag. So sometime we have to go somewhere 100K x magnification or 120x magnification. That will help us to visualize those small protein, and sometime you want to see that okay, there is some iron nanoparticle which is surrounded by sucrose molecule or some kind of this molecule which is using for drug discovery. So those we have to go to very high magnification. Now you can see, when I started I told you the one limit for the light microscope is that magnification, we cannot go to a very high magnification and we cannot reach that high resolution. But the electron microscope, that is the that is the good thing for the electron microscope, where we can go to a very low mag, even a 50x magnification, to a very very high mag. So even you can go 150K magnification to image a protein sample. So generally for the structural biology purposes or some imaging purposes, within 120K magnification or 150K magnification and is good enough to visualize most of the biological sample so far. If you look at this diagram here, you can see this is the magnification we are talking about, and this is a very important part, that is called the dose. When electron dose it will go through, you can see a very bright light here, circular light. I am going to show you those things, and whenever we will go for that workshop, that 6 classes, there I will describe all of this beam, how this beam should look like. So beam will be a circular beam and it is a very bright white beam. Now so when we have that white beam, it will show some kind of dose also, that how much electron dose is there. For the material science sample, the dose is not that important. For the biological science sample, it is a very important part, because if we have a very huge dose, it is going to damage our sample. So we have to regulate that. So this is called the focus. Here we mostly use the defocus term. Why we are using defocus term? I will describe these things in the later stage. But some kind of focus is required here. Now if you look at here, if you look at this panel, you can see what is the magnification, what is your electron dose, and what is your defocus value. Now if you look at here, you can see the high tension, And also what is the spot size? I told you the spot size connected with your condenser lens. What is the current of that objective lens, or objective lens is here. So what is the current for that condenser lens 1, condenser lens 2 and objective lens? So those parameters are not very important at this moment. What is required here? I'll just mark those things. The most important part is your high tension, your spot size, your focus step. Focus step means that how fast you want to do your focusing. So if you just use the focus step 1, you will do the fine focusing. If you just use the focus step 4 or 5, you will change the in a micrometer range very fast, and your focus will be not that fine focusing. So the focus step is dictating what kind of focus do you want. So you should pay attention for the fine focusing. Most of the time it will be better when you will do the focusing, you decrease the size to 1 or 2. And now the another important part is the defocus and your dose and your magnification. Now here also you can see the same defocus value here, and this tells you that camera condition, that whatever imaging condition you are using, it is stable or not stable. So now here there is other parameters are also there. That is also very important. Here we can see x, y and z. What is that x, y and z? So if you just consider this is your entire area where you want to image. So you can move around in any direction. So if this is your sample area and this is the trajectory how you are moving. So in this movement, this may be x and y direction movement. Okay, that I will show you in the next slide how this part looks like. And then there is a other parameter, it is called the z height. So if you remember, when we are talking about that z position or z height, it is a very important point, because that is basically eccentric height of your sample. So it should go in a particular position of your objective lens. So if this is your objective lens, it will go in a particular position and stay there. That is the actual eccentric height. If it will change the position from your eccentric height, it will change the focus of your sample. That is the reason that z height means that is it in your eccentric height or not. If it is not, you should change the position of your sample holder tip and keep it in a eccentric height. Now we will go to the next slide where you can see those are the enlarged view. Here you can see this is the enlarged view of this particular window where you can see all the status. What is the vacuum here? If the vacuum is poor here, it will be better not to do any operation. Nowadays all the systems are very upgraded system, and this is a very sophisticated system I can say. And this sophisticated system automatically, if the vacuum will increase, it will lock the system. You cannot do anything. You cannot open the column valve, you cannot see the beam, you cannot image. So this is the sophisticated system and auto regulated. Now you can see this is the high tension, this is a 200 kV, and all the details here I already discussed about this one. Now we will go to the next slide. So here if you look at the top, you can see there are some options are there. Setup is the first page, and then there is a autoloader system. So that autoloader we are going to discuss in the later stage. So at this moment we are not discuss about this particular part. Only part you just pay attention that when we are doing cryo-electron microscopy, cryo-electron microscopy means we are doing imaging at cryogenic temperature. Cryogenic temperature means it is liquid nitrogen. So liquid nitrogen means it is minus 196 temperature. So if we want to do imaging in cryogenic temperature, your microscope and rest of the part should be in a liquid nitrogen temperature. Here it is showing you the temperature of the different component of this, the docker, holder, cassette gripper, what those are. We are going to discuss that, but you just pay attention to this, that most of the time the temperature it is below minus 180 when you will do the cryo imaging. That is the major difference from the room temperature microscope, or if you just say normal temp. or transmission electron microscope and cryo-electron microscope. In cryo-electron microscope also we are using that transmitted electron beam, but the temperature and sample is maintained within this temperature. So, it is a electron beam is not at cryogenic temperature, electron beam is as it is. So, only the sample holder region we are keeping a very low temperature. Okay. Now, this is the trajectory I am talking about when we are talking about that imaging, that okay, your sample area it is looking like this. So, this is called the entire sample area or grid, and now this is the trajectory that I am going in a different direction and selecting that which area I want to image at high magnification. So, you can follow any trajectory, no one is stopping. Here I am showing you one of that trajectory where you can see in a very smooth way initial pattern it is going like this. So, it is following some kind of pattern, and initially we are making our atlas and checking the entire grid. When this is done, we are just moving around randomly to select some area. Here it is showing you the beam setting, what kind of spot size we are using. We are using spot size 7 here. So, why we are using the spot size 7? Because the spot size 7 it is going to reduce the electron beam. For the biological science sample, this is very important. Here you can see there is a one parameter, it is written Obler. This Obler is basically helping us to adjust the eccentric height. When you will do the practical class or the workshop, that time we will discuss about all of these things in details. One more important part, that is the control panel. I discuss about your intensity button, intensity. I discuss about the focus, where those are. There will be a two control panel in a left hand side and a right hand side. So, if you look at this, you can see there is a joystick, then there is a eccentric height. I already discussed that eccentric height and Obler, this is going to help us to adjust that Z height, the plus and minus, it can go up and down when you are talking about plus and minus. Now, this is our focus button which is helping us to focus the sample. Here we can see the magnification, which is the intermediate lens and the projector lens. Focus is our objective lens. Now, you can see the intensity, which is our condenser lens 2. And then beam shift, we want to move the image from one position to other position. We are using this joystick, X and Y joystick, and if we want to shift the beam within the screen, we can use this beam shift X and Y. If we want a tilt angle, that we can also regulate it, and also there are few extra buttons are there. Whatever options we want to include here, we can include here. So, these are the major options. Then I told you that multifunction X and Y is the another point where you can see this multifunction X and Y. This multifunction X and Y is a very important tool in our microscope, because this is the tool, it is connected with all the lens system whenever it is required to condenser lens or objective lens, and this tool we are using to correct different kind of aberration, mainly for astigmatism, condenser astigmatism, objective astigmatism, or if we want to align our electron beam, like okay, we want to correct the gun tilt, gun shift, then beam tilt, beam shift. All the cases this multifunction X and Y is a very important parameter, very important tool which is going to help us to align the beam. And the multifunction X and Y is connected, mainly connected with the quadrupole lens system, because quadrupole lens I already discussed about that, it is a north south it is connected. This quadrupole lens system is connected with our multifunction X and Y. So, this is the overall architecture of that any electron microscope. If you go to the electron microscope, those are the common things you can see it in any microscope facility. So, I discuss about the user interface, I discuss about the control panel also. So those knobs are very delicate. Whenever anyone will enter the microscope room, you should be very careful. You should not touch all of this button or the interface, because if there is a certain change with those intensity or the spot size or the focus, it will take a lot of time to bring it back. So this is a very delicate instrument, so you should be very careful when you will enter into the into any electron microscope facility. So I will stop it here today, and the next class we are going to discuss about the ray diagram and how we can get a magnified image or high resolution image or low resolution image. Thank you all for your attention.